I'm going to start off with kind of like a big overview and then dive a little bit deeper into some of the specifics on the science side. But just in case you have a hard time remembering, I feel like some of these terms are kind of hard to remember, but SNPs is just single nucleotide polymorphism, which basically means polymorphism means that, it could be changed between any of the 4 nucleotides. It's just a change out of a single nucleotide. And why this matters really comes down to what happens with that single nucleotide. So here's my analogy for the day. Think of a car assembly plant. So if they're assembling these cars and they're following this recipe or this blueprint, and if the blueprint is messed up, and normally this car would need this kind of bolt to hold the frame together, but they grab a different bolt, right? It's a different bolt. Or maybe it doesn't matter. Maybe it makes no impact on the car. Maybe a different taillight, maybe a different, part, they can interchange it and it's not a problem. But if there's a part on that car that they interchange and now you can't shut the door, you can't start the engine, you can't use the brakes, suddenly it's really important. And that one change can impact the function, can impact your ability to use the car. And so the idea here is the SNPs we care about are the ones that impact function. And we're going to kind of talk about, in the protein, kind of getting to not just a car assembly now, but thinking about the protein assembly in our bodies for all these different proteins, how these SNPs are, that blueprint is calling for a different part for that particular spot. Do I care? Is it affecting my function of that protein? And how does that play into that system that protein operates in? So just as a refresher, because even as a scientist, I'm like, oh, that's right. DNA to RNA first, that happens in the nucleus, that's transcription. Then the RNA heads out of the nucleus and then the ribosomes take over and convert that RNA to protein. And of course, we're hearing a lot about RNA now with the vaccines that are messenger RNA. So I think that was kind of a helpful reminder to me. But, again, a change in just one nucleotide in that DNA blueprint affects the RNA, affects the final protein. And just a few kind of reminders, I think, for me that are helpful. So DNA is that double helix in the nucleus, that blueprint with the 4 nucleotide code. And what I think is so amazing, the whole biology of this is how can you compact all of that DNA into these chromosomes? It's compacted 10,000 fold. So, if you stretch it out versus you squish it up. Whereas RNA, right, that's just a single strand that's made from the coding strand of the DNA. The 4 nucleotide code is similar, but now the thymine is uracil. So you've got one switch out of one of the bases, and it's very unstable and temporary. It gets degraded in the cytoplasm. So our nucleus, we need to keep making RNA to keep making that protein from that DNA template. Ooh, bless you.
Oh, excuse me. I'm sorry, I was muted. I'm getting a cold, which is why you have a stock photo of me, because it's not my morning. Sorry, I thought I was
totally get that. And then finally, the RNA, right? The ribosome comes, reads the RNA, makes that protein. Proteins are large. They are complex. They have to fold an exact certain way. There's all these other proteins called chaperones or chaperonins that help that protein fold appropriately. There's 20 different amino acids that that RNA can code for. And then the function could be structural, kind of like collagen. It could be part of our signaling pathway. It could be, could bind and bind other proteins and activate them or suppress them. It can bind to DNA and activate DNA transcription or shut it down. It can be an enzyme. It actually does chemistry, right? The whole, like we hear about the dehydrogenases in the Krebs cycle. So just a little review of that science. And then I thought, let's dive into each one of these just a little bit more. So DNA, we've got our adenine, our guanine, our thymine, and cytosine. And this idea that these pair up specifically, right? The T and A always match up, the C and G always match up. And they match up through very weak hydrogen bonds. But that matchup is what kind of holds the DNA, that matchup together. And if something gets methylated or changes and there's a bulge in that DNA, that then says, oh, we have a repair. We got to get these repair enzymes in there to take a look and see if we need to fix something. And so based on kind of just thinking about what that nucleotide looks like, Now, when I look at a double helix, instead of seeing the ribbons and the ladder look, I'm realizing that the outside structure really is the phosphates and the sugar part, and the inside part are the actual nucleotides that are base pairing up. So I kind of like this structure that to me is a little bit more realistic. But here's some stats that, again, when I start thinking about this, I am just amazed at what our body is able to do. I mean, here we have 3 billion DNA base pairs in the human genome. And I'll never forget, in my job when, like around 2000, when we're sequencing the human genome, and I was in a company at the time, we were doing a lot of supplies of products for, companies that were into sequencing and looking at this stuff. And they're like, oh my gosh, there's so many base pairs. We're going to have 100,000 genes in the human genome. So when it came out that we only had about 20,000 genes, everyone is like, what the heck is up with that? We're so complex. There's simple worms that have way more genes than we do. And it turns out that about 98% of our DNA is not even coded for proteins. And it's still a mystery to this day. They keep finding out that, yeah, those little, the little pieces in between the coding segments, they are actually important. They are important for signaling. They're important. They interact with proteins and enzymes. They interact with other RNAs and DNA. It's like crazy stuff. So here we thought we had this system figured out. It's like, oh, it's the protein that does the business, right? The protein that actually matters. And now we're finding out those RNAs that don't ever code for proteins are super important. And there's so much we don't yet understand about that. And again, this is a, we have 4 base pairs in our RNA to code for the protein. So this is a digital code, kind of like with, you think about computers where it's ones and zeros. Here we have 4 codes, and these 4 codes occur in different codons, right, in the RNA that then code for different proteins. And just a simple replacement in one of those nucleotides, like I have circled in red, there's a UCU. Well, that would normally bring in a serine. But if that first uracil changes to a cytosine, now it's going to bring in a proline. And maybe it doesn't matter, but maybe it does. Maybe it is the business part of the protein. Or here's a UAU for tyrosine, but if that last U gets changed to an A, now you got a stop codon, and it's going to cleave that and terminate that protein before it's finished making it. So, it's amazing to me how they say how our DNA is constantly having breaks, but those DNA repair enzymes, they are so busy and so important to kind of keep everything in check. But we have lots of SNPs that we don't care about because they don't impact it. And again, just to geek out a little bit more, I thought, well, I'm going to throw up a slide just so people remember a little bit about these 20 amino acids, because some of these are charged, they're acidic. Some of these are bulky and big, and some of these are, basic. So just thinking about, if you switch out one of these for a different one, it can change the shape of that protein, change its ability to do its business, to bind, to signal. And it could maybe just drop that a little bit, or it could totally get rid of its function altogether. And then I thought it was important to think about the heterozygous versus the homozygous, because especially when we start looking at the functional genome stuff, the functional genomics information, just a reminder, we get one set of our chromosomes from our mom, one set from our dad. And if that gene that has that SNP is just on one of those sets, it is heterozygous. But if it somehow ends up showing up in both of them, if you get that kind of nasty SNP from both your mom and dad and now it's homozygous, you have even less, right? You have, potentially even less activity or function, or maybe none at all, of that particular protein. And if you have a question, just feel free to pop in here. I'm just about done. And then I want to end with like a little 2.5 minute animated video that I found that like, ooh, this looks cool. They kind of put this whole thing together in one little cartoon.
Tina?
Yeah.
Would you mind just going over again the heterozygous versus the homozygous? And if it's not one or the other, is it always going to be one or the other, or is it sometimes neither?
Thank you. Yeah, for asking that. So for example, if you have the wild type, so wild type is the neither. If you have the kind of the common normal amino acid / nucleotide base in that position, it's called wild type. So I should have put that up here too. But if you have one of your chromosomes that you got maybe from mom that has that one SNP, maybe in your COMT or your MTHF, what is it? Anyway, your methylating gene, then it's heterozygous. But if you were super unlucky in the whole lottery thing and you've got 2 bad SNPs, so you got both chromosomes having both bad SNPs, now you have no backup, no extra copy that can make the good functional protein. Does that make sense?
It does, thank you.
Julia, and Tina, maybe you're going to cover this, but excuse me. This really comes up a lot when people are looking at MTHFR, which they've associated with a lot of the clinical conditions, depression, anxiety, clearing estrogen. So on the Dutch and on the nutrition genome, the 2 main ones you want to target in on when you're trying to help someone understand estrogen metabolism is MTHFR and COMT, COMT, which is very specific to estrogen, dopamine, some of the more steroid type metabolites. And there's a lot of other things on the nutrition genome, but when I talk to people around hormone mediated cancers, those are the 2. And this is a funny thing, and maybe it'll help you. I remember when I was first learning about this one woman was making a joke, and she came out and she's like, "I'm coming out as homo," and I was, and then she's like, "I'm homozygous." But when I think about like the meaning of this, homozygous just is like, when I tell people, if you've got a heterozygous COMT, depending on your nutrients and lifestyle and all these things that we talk about, you may or may not be showing clinical symptoms of it. But if you come out as homozygous, you basically are dependent on all those nutrients and lifestyle changes to give you that support, because you're not getting any from your genome. So homozygous is a more severe, when we correlate this to clinical presentation, the homozygous people, particularly in COMT, that's where you really have to hit some of the supplements, magnesium and those kind of things, because they're not getting any support. So homozygous is more serious, but the 2, especially from a breast cancer or hormone level you want to look at, are MTHR and COMT.
And if I can add to that, the book Dirty Genes by Dr. Ben Lynch is fabulous at cleanly explaining so much of this. And he goes through 7 of the most common SNPs and how they manifest. So even like COMT, which I'm heterozygously positive for, can manifest either in a slow fashion or a fast fashion, meaning how fast does your body push things out or hold them back. I think I'm fast COMT from reading that. It's really kind of interesting. So if you haven't read that book, definitely add it to your list. It's really good. The easiest way to understand the heterozygous and the homozygous is exactly as Deborah was saying, heterosexual, male, female, homosexual, female, female, male, male. So we have the same, or we have one of each. And the one of each comes from, did we get a dirty gene from our mom, but not our dad? Or did we get a dirty gene from our mom and a dirty gene from our dad? Or did we get normal genes from both parents, in which case we're wild type? So that's how I kind of make sense of it all.
Thank you. Yeah, I appreciate you applying it from the clinical or medical perspective, because my background is science, not thinking about it in those terms. So thank you for that. And I think, the other thing to remember is that this whole idea of really knowing what role SNPs play in different, in different symptoms, basically the phenotype, how does that show up? We have so much to learn. We are just at the tip of the iceberg of understanding SNPs. And then, as I was just mentioning, now we better understand that proteins are just a small piece of the bigger signaling puzzle. So after this, or at the same time as this, people are studying microRNAs and long long coding or non-coding RNAs. And it's just, as a scientist, I feel like the more I learn, the more I realize how much we don't know and the more we have to know. But let's use what we know. So just about done here, just a couple other last things is, so again, the way these proteins fold, it's amazing. These things are so complicated. They have helixes and beta sheets and turns, and the way that they fold and interact with, if there's a big bulky amino acid or a charged amino acid or a sulfur there. I mean, just like curly hair, right? That's where your amino acids, you've got sulfurs that are locking together to hold that hair so it's curly. Anyway, here's the human growth hormone polypeptide. This is a smaller hormone, but a lot of times these proteins form complexes with themselves and other proteins to form the functional counterpart. But just to give you an idea of, here's just a diagram of like how they form the helices and these basic turns. And here's pancreatic lipase, which of course, digestive enzymes are so important if we're going to get the nutrients from our food. And pancreatic lipase is kind of a big one. But here again, looking at how complicated and big these proteins can be. And then the whole idea that these things play into all that signaling. Like this is just T cell receptor signaling, and these pathways, they can be so complex and redundant. So again, we have a lot to learn, and we've learned a ton, and we're tapping into that. But the science is going to keep changing. This isn't something we can just walk away and say, oh, we've got this figured out. We have to keep coming back and getting updates on, like the nutritional genome. They're constantly updating and adding in more SNPs as we learn more. So just this idea that our bodies are amazing and they're incredibly complex. And if one of these things, like in the signaling pathway, gets a little overactive or a little underactive, you can imagine how everything, then it's a domino effect, everything gets impacted. And how can we then go in and support these signaling pathways? So with that, I would like to show you, if I could figure out how to show you